sex determination. In sexually reproducing organisms, meiosis creates both genetic variability and ensures genetic constancy. This depends on fertilization, which relies on some form of sexual differentiation, that is, presence of male and female individuals. Sexual differentiation is achieved by a variety of sex determining mechanisms. The genetic basis of sexual differentiation is usually related to different chromosome composition of the two sexes. The heterogametic sex either lacks one chromosome or contains a unique heteromorphic, which is different in size and shape, chromosome, usually referred to as the Y chromosome. In humans, the study of individuals with altered sex chromosome compositions like Klinefelter, Turner syndrome, this has established that the Y chromosome is responsible for male differentiation. The absence of Y chromosome leads to female differentiation. On the other hand, in Drosophila, the Y chromosome has no such role. Instead, a balance or ratio between the number of X chromosomes and the sets of autosomes decides the outcome. Autosomes are the chromosomes other than the sex chromosomes. In humans, these are 22 pairs. Sex determination in humans. A normal human has 23 pairs of chromosomes, out of which one pair varies in configuration between the male and the female. This pair is known as the sex chromosome. Designated as the X and the Y chromosome, the human female has 44 autosomes and 2 X chromosomes, and the human male has 44 autosomes and 1 X and a Y chromosome. The eggs produced by human female during oogenesis has 22 autosomes plus 1 X chromosome, whereas the sperm produced by a human male has 22 autosomes and either 1 X or 1 Y chromosome. Eggs fertilized with a sperm containing Y chromosome result in a zygote that develops into a male fetus, whereas if the egg is fertilized by a sperm having a X chromosome, it will lead to development of a female fetus. Sexual differentiation. During the early stages of development, the human embryo is neither male or female. By the 5th week of gestation, the gonadal cortex and the gonadal medulla are formed. The cortex is capable of developing into an ovary, while the medulla may develop into a testes. If the cells have XY constitution, the gonadal medulla develops into a testes. This takes place around the 7th week. However, in the absence of Y chromosome, no male development takes place and the gonadal cortex subsequently forms the ovarian tissue. Thus, the Y chromosome is responsible for male differentiation, and the absence of Y leads to female differentiation. The Y chromosome The Y chromosome consists of a gene or genetic information that provides the signal responsible for sex determination. The triggering of undifferentiated gonadal tissue of the embryo to form the testes is caused by TDF, or the testes determining factor. Euchromatic region contains functional genes and the heterochromatic region lacks genes. Dosage compensation. Dosage compensation prevents excessive expression of X-linked genes in human females. Since the females have two copies of the X chromosome and male have only one, there is a potential for females to produce twice or double the amount of each gene product associated with the X chromosome. The normalization is achieved by inactivation of one X chromosome, either of maternal or paternal origin, during the early stages of development. This process, or normalization, that is inactivation, results in the formation of Barr bodies in female somatic cells. Barr bodies. Barr body can be easily demonstrated in human female cells. It is a highly condensed structure and lies against the nuclear envelope of interphase cells. Barr body is also known as sex chromatin body. It is inactivated X chromosome. Regardless of how many X chromosomes exist in different individual cases, all additional X chromosomes are inactivated and appear as Barr bodies, except leaving a single or one copy of the X chromosome. Therefore, the number of Barr body follows a n-1 rule, where n is the total number of X chromosomes present. In Turner syndrome, as there is only a single X present, no Barr bodies are seen in somatic cells, whereas in Klinefelter, there is an additional X chromosome, thus one Barr body would be observed. In females, where there are 3 copies of X chromosome present, 2 Barr bodies would be seen in somatic cells, leaving a single X to continue functioning. These Barr bodies can be used to distinguish between normal male and female cells. They are useful for diagnosing various kinds of abnormalities related to sex chromosomes. The Lyon hypothesis. In human females, one of the X chromosome is of maternal origin, while the other is of paternal origin. Now the question arises is, which one of the X chromosome is inactivated? Does this inactivation occurs randomly? And whether the X chromosomes of similar origin are inactivated in all the somatic cells of an individual. Whether the same X is inactivated in all the cells, these answers are explained by the Lyon hypothesis given by British geneticist Mary Lyon in the year 1961. The inactivation of X chromosome occurs randomly in the somatic cells at an early embryonic developmental stage. In human female embryos, sex chromatin bodies, that is the Barr bodies, are observed by the 16th day of gestation. Once the inactivation has occurred, all the progeny cells arising from that single cell will have the same inactive X chromosome. Further, females are mosaics where cells in some portion of the body express paternal X chromosome, while the cells in some other part of the body might be expressing the maternal X chromosome. Sex determination in Drosophila. Similar to humans, Drosophila also shows same composition of sex chromosomes, that is male has XY chromosome and female has two X chromosomes. Here the Y chromosome is not responsible for maleness in flies. This has been shown by the work of Kelvin Bridges in the year 1916. Bridges proposed that instead of the Y chromosome, both the X chromosomes and the rest of the autosomes together are responsible for the sex determination in fruit fly, that is Drosophila. Bridges' work was based on non-disjunction of X chromosome in females. And subsequent fertilization by sperm of a normal diploid wild-type male. Studies on the resulting progeny was also done. Non-disjunction is the failure of paired chromosomes to separate during anaphase of either the first or the second meiotic division, which results in production of abnormal gametes. In Drosophila, one set of autosome consists of 3 chromosomes. Here, the second, third, and the fourth. The chromosome first is the X chromosome. Presence of one X chromosome and 2 sets of autosomes produces a normal diploid male Drosophila. X is to autosome ratio would be equal to 0.5, whereas presence of 2 X chromosomes together with 2 sets of autosomal chromosomes produces a normal haploid female Drosophila. X is to autosomal ratio would be equal to 1. This mode of sex determination is explained by the genic balance theory. Sex determination in birds. In species of birds, sex determination is identical to the XX-XY mechanism, but here the females are heterogametic and the males are homogametic. In birds, the chromosome composition of the egg determines the sex of the offspring, whereas in humans and in fruit flies, the chromosome composition of the sperm determines the sex of the offspring.